Good morning, class 11th. Today we will go for the first kingdom, Kingdom Monera, which is in your work chapter 2.1, Kingdom Monera. Dear student, you already know that the general characteristics features of the Monera, that they are prokaryotic, primitive type cells, and they have unicellular and prokaryotic microscopic. All bacteria and viruses are included in this group. Dear student, the number of comments are given in your book. They are microorganisms, abundant microorganisms, and they are growing on the desert region or hot spring. Extreme habitat also they can grow in the ice region. Everywhere they can grow, and on the basis of their shape and size, they are categorized into many types: coccus, spirillum, comma shape, and rod shape, bacillus, spiral shape, spirillum. So, bacterial structure is very simple, and but you know that their behavior is very complex with the cover compared with the another organism. And bacteria is a group which show most extensive metabolic activities, and some bacteria are autotrophic and they synthesize their food from the inorganic substrate. They may be photosynthetic autotrophic, chemosynthetic autotrophic, and vast variety of bacteria are heterotrophic. Heterotrophic means they do not synthesize their own food. They are depend on the others organisms. They are depend on the dead and decaying matter like saprophytes for their food. So, dear student, the number of characters are given in your book, and you know the cell wall is present, which is made up of peptidoglycan or polysaccharides, and they are autotrophic also, heterotrophic also, just like I explained you before that, and the macroscopic 0.1 mu micron in length, very small size. Okay, and they have no well organized nucleus. The nuclear materials are scattered in the center of the cell, and there is no true nucleus. There is no cell membrane bonded structures like mitochondria, Golgi body, and endoplazmic. So, dear student, these are the common characteristics features, and you know that uh, the classification is given here. That is first one is Archaebacteria and second is Eubacteria. Dear student, Archaebacteria, they have cell wall which is made up of protein and non-cellulosic polysaccharides. The cell wall consists of that. They have no peptidoglycan in their cell wall, and branch chain lipid are also found in archaebacterial cell wall, and see they are sensitive for the antibiotic. In eubacteria and archaebacteria there is differences for that. Habitat very much similar to primitive environment of the earth. Extreme condition they can survive, and that's why the archaebacteria is regarded as the fossils, living fossils. Okay, and the habitat which is very similar to the primitive environment means some you know that they can grow in the acid region, so they are called as acidophiles. They can grow in the methane area where another organism cannot grow, that is called halophiles. And you know they can tolerate the extreme condition of temperature, so that they can also classify the third group, thermoacidophile. Okay, so dear student, archaebacteria are able they are able to produce the methane gas, so they are called as methanogens. Okay, on the basis of the physiological behavior, archaebacteria are classified into three groups. The first is methanogens, means they produce methane, and you know that they are also called as anaerobic. They form methane and from the carbon dioxide or the formic acid, and they are also known as methane producers. And you know that bacteria is also found in the rumen of the cattle. Rumen is the pouch of the stomach of the cattle which are herbivores, and they are help in the digestion of cellulose of the cattle, because they are taking cellulose in their diet. Methanogens are also responsible for the production of biogas, and example that is methanobacteria and methanococcus. These are the examples. Second one, that is halophiles. Second one is halophiles. Dear student, halophiles means they are salt loving. They are This group is contain anaerobic bacteria, extremely brine salt solution. They are grown there, so they are called as salt loving. Example: halobacterium, halo, um, coccus, yeah, haplococcus. So, dear student, in strong light, they have a pigment membrane, purple membrane, and they can utilize the solar energy to run their metabolic activity by producing the ATP or energy molecule. ATP, adenosine triphosphate. Now, thermoacidophiles. Third is thermoacidophiles. Dear student, thermo means temperature, acido means acid. So third, that is, it contains aerobic as well as anaerobic bacteria, and they are found in the hot sulfur spring region, and these act as the high temperature, that is 80-degree temperature they can survive, extreme condition they can survive. Okay, and we see that because of their special nature, the extreme environment habitat they can tolerate, or the archaebacteria appears to be the ancient time. Uh, to be an ancient time, because it live in such environment which resemble with the primitive type environment of the earth or primitive condition of the earth, so that the archaebacteria are regarded as living fossils. They can tolerate the extreme condition of the environment. So, dear student, this is the bacteria, archaebacteria. Second, eubacteria. This is also called as the true bacteria. What it is? True bacteria. And dear student, true bacteria, same characters are there. In your book, it is written here that thousands of the different true bacteria are there, characterized that they have rigid cell wall, they are motile, they have flagellum, they are also called as cyanobacteria, which is called as the blue-green algae. They are found in the pond, or you can say that field where the paddy crops is growing. They are photosynthetic autotrophs, and they may be in unicellular, they may be in colony, they may be filamentous, their body structure. The colony, they have mucilaginous sheath around them, and they form the bloom in polluted water bodies. Some of the ways they can fix the atmospheric nitrogen from their heterocyst, very specific characters in them. Heterocyst, you know that previous class I explained you like this structure, and this is the structure of anabaena. And in some cells, they are forming the thickened structure that is called as heterocyst. It is called as heterocyst. And this heterocyst is help them to fix the atmospheric nitrogen and increase the productivity in the rice field. And this is the name anabaena, azolla. This filament Podogonium, these are the true bacteria or cyanobacteria, you can say. Dear student, they perform photosynthesis, so they are called as photosynthetic bacteria. Some of them, they are synthesize chemicals, so they are also known as chemosynthetic bacteria, sulfur bacteria, hydrogen bacteria, and heterotrophic bacteria. The most of them in the abundant in the nature, and they are most important decomposers when the dead bodies of the plants and animals remain in the open. These heterotrophic bacteria grow on them, and they release the enzyme, and after the releasing enzyme, they split the complex molecules or insoluble molecules of the dead bodies into simpler soluble form. And in this way, they decompose the dead body and mix them into biogeochemical cycle. So they are help in recycling. They also help in the making of useful things such as curd, cheese by the milk, And lactogenic bacteria, they are change the milk into curd. It is useful also. Some are useful for the production of antibiotics, some are useful for the fixation of nitrogen, and some are dangerous also, that they are cause of the disease. They are disease causing. When bacteria are disease causing, they are called as pathogens. They are pathogens in the human, plants, crop, or the farm animals, and the pets also too. Cholera, typhoid, tetanus, citrus canker in the citrus fruits, and number of vibrio cholerae, pneumonia, leprosy. These all are the disease which is caused by this bacteria, and so that they are called as pathogenic bacteria. The reproduction take place in the bacteria, that is asexual, sexual, or vegetative. Vegetative reproduction is done by the binary fusion, and asexual reproduction is done by the formation of endospore, and sexual reproduction is also there by the conjugation, transformation, and transduction. These are the method for the sexual reproduction, where the DNA from one bacteria transferred to the another bacteria. Mycoplasma is there. Mycoplasma is there, which is the most smallest organism in the world, and they have lack of cell wall, smallest living cell. They are survive without oxygen, and many mycoplasma are pathogenic in the animals and plants and human. Animals and plants and human also, they make the human infertile. So this is the kingdom Monera's characters and classification. But in the NCERT book, this explanation is given, but I will explain a little bit more. So, dear student, you know that Monera and single cell I am drawing here, that this is the capsule, after that the cell wall, and then cell membrane, and cell membrane form the lysosome. Nuclear materials are scattered in the center, there is no nuclei, that is called ribosomes, polyribosomes like this, and fat free ribosomes and RNA are there, and cytoplasm and membrane also, flagellum also there. This is bacteria. What is this? Bacteria or mammalian cells. So you labeled it capsule, cell wall, cell membrane, RNA, DNA, nucleic acid, polyribosomes, flagellum, cytoplasm. These all are the structure I already explained. Okay. So dear student, you learn it, and later on I will explain a little bit more. Thank you.